Good afternoon, ladies and gentlemen. I would like to express my gratitude to the organizers for inviting me. Could you get closer to the microphone and not to speed up too fast? We have this simultaneous interpretation provided. Thank you. Yes, I understand. All right, so perhaps I will switch off my camera. So the first key steps in implementing internal dosimetry for patients in the clinical nuclear medicine department is the topic of my lecture. This is the plan of my presentation. Next slide, please. I cannot do it myself. So first of all, let me remind you what internal dosimetry is. Then I would like to speak about the current status of and prospects of internal dosimetry in individualized therapeutic planning. Then the third point is the main limitations that are found in implementation of internal dosimetry in clinical practice, how to choose the appropriate tool for calculating absorbed doses in patients, and last but not least, calibration and validation of tools for internal dosimetry, which is based on SPECT, CT, and PET-CT image analysis. Next slide, please. Internal dosimetry in nuclear medicine, this is the field that deals with the assessing of the dose of ionizing radiation, which is absorbed by internal tissues and organs due to administration of radiopharmaceuticals. It involves modeling biodistribution, also calculating of the energy distribution of radiation and evaluating potential biological effects, which is really important in radionuclide therapy and radiological risk assessment. Nuclear medicine for therapeutic purposes, here termed molecular radiotherapy, MRT, is a rapidly developing field, mostly in the context of metastatic cancer treatment. Here you can see the picture that shows the series of PET images after application of tototake. This is the use of alpha emitter. Here, the therapy and monitoring was performed in Germany, in Heidelberg Universal Hospital. Next slide. The implementation of radiation dosimetry in routine clinical practice faces different challenges that may introduce unprecedented opportunities for cancer treatment. Here you can see the publication that provides the stance of the important institution, and this publication of December 2023, you can find the general guidelines. And here you can see the image of a computer with the use of Hermia software that is applied for internal dosimetry. The program of nuclear medicine and diagnostics made it possible for laboratory of in Szczecin to perform such an internal dosimetry. Here you can see Edwin McQueen, a physicist that helped us to calibrate the Hermes system with our gamma cameras. And due to very close cooperation with Hermes service, dosimetry, internal dosimetry is not a very difficult field. We are using Hermes dosimetry software right now, although we have also other software. This picture comes from internet, is published on internet, and you can also find some pictures, other videos on YouTube. Next slide. Here in this slide, you can see a picture of an internal dosimetry laboratory. So one, dosimetry physics that deals with the dosimetry that has a lot of things, and a kind of a mess, but it is a controlled mess. An expert in the field of clinical internal dosimetry, Manuel Bardies, in his publication, described the current state of dosimetry. And he said that this field is currently caught in a vicious circle because we have insufficient evidence for clinical benefit of dosimetry, and this has supported arguments against its implementation, which in turn have prevented the accumulation of evidence. So actually it treats this type of dosimetry, internal dosimetry, as chemotherapy. And actually, we have a proper dose of active substance determined that we should follow. However, there is more and more evidence that shows in the research that there is a correlation between the absorbed dose and the clinical effect, treatment effect, which shows that dosimetry definitely is very beneficial for the patients. Next slide. So why there are so difficulties in introducing internal dosimetry to nuclear medicine? First of all, there are technical problems. So first of all, dosimetric imaging and analysis is not easy. There is still no general recommendation how to perform the calibration quantification of such a dosimetry, and then integrated accessible software is lacking, and actually a patient needs to be imaged multiple times. We have 2 gamma camera SPECT CT and multiple scanning of a patient is very time consuming. And last but not least, regarding technical causes, well, the accuracy is still questionable. There are also some other problems that internal dosimetry is facing. This is the shortage of medical physicists that are trained, and some of the therapy radionuclides are difficult to image. For instance, the distribution of alpha emitters in the body of the patients is very hard to be obtained. And last but not least is the lack of awareness among healthcare professionals that individual planning of radioisotope therapies really increase the effectiveness.
CERT with microforts is currently the only radionuclide therapy modality where dosimetry has a proven benefit in terms of clinical outcomes, that is, improvement of the patient's health. SIRT, selective internal radiation therapy, is a modern modality to treat tumors, in particular liver tumors. So radiation is administered using microspheres directly to the organ. So spheres are used and they are filled with radiative isotope. It produces beta radiation, which provides very precise radiation to the tumor cells, and the impact to healthy tissues is minimum. So microspores are introduced to the tumor, and the vessels provide the agent. The expert wrote, "It is time to change this vicious circle of clinical internal dosimetry. It should be turned into the circle which will treat patients. Dosimetry implemented from the start of radiopharmaceutical development will accumulate results that can in turn be used to optimize patient management. They can be used to optimize therapies for the patients. Therefore, this is the best time to implement clinical dosimetry in clinical nuclear medicine departments. It is time to secure funding for development of internal dosimetry, to purchase commercial programs for clinical dosimetry, or create our own programs to calculate absorbed doses. It's good time to publish our results of measurements and therapeutic effects. Euratom Directive of 2013, it states that the radiotherapeutical dose should be planned individually. So when this directive was adopted throughout the European Union, there are unprecedented options to develop very conscious way of treatment that will mitigate the risk of insufficient or treatment or too much treatment. Medical physicists are not wasting time. A recent call by the European Federation of Organizations for Medical Physics in their Medical Physics journal, and it states about some arguments to have some clinical practice in terms of internal dosimetry. So there are 14 postulates, and I'm just quoting the first 5, which to me are most important. I would like to encourage you to reading these postulates, recommendations. Item number 2. We need national and European databases to collect data on clinical factors, dosimetry, and patient outcomes from different centers. The red labeled recommendation, imaging and patients' dosimetry must be reimbursed, as it is in the case for external beam radiotherapy. Next slide, please. Let's assume that the specific nuclear medicine center has managed to influence the decision makers to implement internal dosimetry for clinical practice. So what will be the first steps of the physicists? What should they start with? First of all, they need to select the dosimetry programs. There are lots of such programs. Sometimes people ask me which program should be purchased. Then I answer that by these programs that you understand very well, you shouldn't use the black boxes. The next step is the calibration and harmonization of the apparatus used. So sensitivity, measurements, looking at the counts of gamma camera, and appropriate data have to be downloaded. Then you have to check whether the dosimetry program reads the information from the gamma camera. Dosimetry reports are very easy to generate. You just need to start the program, computer program, software. But we don't know whether the results, the VOI, the view of interest, is it correct? We need to verify this. To physicists, it's a very easy task. Here in the slide, you can see a cross-section of the NIM phantom. It's filled with lutet. And on the right-hand side, this is the result of the verification, the result of the control, whether the activities are shown in the right way. The activities that have been administered to the sphere. So there are 6 hot spheres, zones. For TechNet, it's very easy to see them in the cross sections. For other radioisotopes, Maybe they cannot be that clear. The biggest sphere is here. The result of recovery coefficient. So the ratio between activity imaged and measured, it reached the value of 70. Next steps for clinical dosimetry after the calibration: acquisition of the series of images for the patient's data input on the activity administered, registration, so positioning of the images, series of images registered. This is a necessary step before we start the quantitative analysis. Segmentation of the regions of interest. It can be time consuming or not necessarily. Here we assess the activity accumulated in the patient's tissues. The next step is to calculate the absorbed dose of ionizing radiation, and then we produce the dosimetry report. Several years ago, for the first time, I generated a dosimetry report. I was learning how to use the dosimetry software when I was quite surprised because this looked like a very serious document, lots of data of the patients, many numbers generated by me, but they were generated accidentally. They were not, if we use dosimetry programs that are not calibrated, they can distort the results. Clinical practice should exclude any accidental data. When we generate the report, we have to be certain on the doses, on the absorbed doses value. So what should we start with to to verify the programs? Before I present our results concerning the first steps, I would like to present a list of the available dosimetry software. This is the global academic and commercial softwares. So I would like to recommend Slicer 3D, developed by a French team headed by Professor Manuel Bartiers. This has some unique options, but it's quite hard to use it, just like academic programs. International Atomic Energy Agency also has also supervises and verifies some academic softwares. Commercial dosimetry software for internal dosimetry. I recommend Hermia software. This company's specialist assist you with launching this software. So we have a dosimetry software. So what should we do next? When it comes to Poland, 4 softwares are most important: MIM, Schupan Hermes, QDose, and Planet Dose do not reconstruct the cross sections independently as the dosimetry programs, softwares. So you need to provide the software with the files, reconstructed files. The first step after launching the dosimetry software is to input the gamma camera sensitivity values into the appropriate protocol in the dosimetry software. The operating manual should be clear on this. Usually we need 3D acquisition of the cylinder filled with the solution of radioisotope for the purpose of studies. You can use Jaszczak phantom cylinder or NEMA cylinder, which is shown at the bottom. We also need to calibrate the sensitivity sensors, but When it comes to the users, well, we have to be careful about it because sometimes the operating manuals formulas are not quite in line with the formulas used in the codes of the software. So you need to use the coefficient of sensitivity. You also have to look at the collimator. So sometimes you will have to change the units, or you have to look at the number of detectors in gamma camera. So the dosimetry programs have to, softwares have to be controlled, and you need to assess the values of the doses absorbed by the patient's tissues. System validation. We have registered a series of NIMA Phantom. So we filled the device with TechNet. And in second experiment, we used LUTET 172. The cylinder of the Phantom was also filled with the radioactive solution, and the concentration of radioisotopes was 8 times lower than in the spheres. The images by the same Phantom were registered by 2 gamma cameras of different manufacturers, and the test lasted 5 days. Validation included verifying whether the activity measured by the specific management will correspond with the values of the activities in the spheres. When we were analyzing the GE images, we had to change the units of concentration of activity into kilobecquerels per milliliters. So initially we didn't obtain, for Q dose, we couldn't obtain appropriate results of the validation. These are the next doses. So the results for MIMA Phantom filled with TechNet were assessed using the software. So this is the image obtained by the gamma camera made by GE company. Measurements of activity in the images were correct. So this is correct curve, correct activities. On the right hand side, this is a failure. So these are the images that were reconstructed by Expect software. So DICOM of the files was not read properly, so we had to download the images reconstructed using the standard software. Despite this change, we still had a problem with selecting the units of concentration of activity in the UDOS software. Effects of the validation for the Hermes system. On the left-hand side, you can see for the results for the NIMA Phantom filled with TechNet. On the right-hand side, results of the experiment performed several months later. Control of accuracy of reflecting activity. So Hermes system performed the reconstruction. So it was successful verification for and for TechNet. So then we cooperated remotely with the service. So we agreed all the details, especially for the collimators introduced into the Hermes system. Which internal dosimetry program is the best? I would like to repeat that this program that you understand properly and you use it with good knowledge. So it will be useful not only for the purpose of science, but also for routine clinical dosimetry. In one screen, you can actually see several windows you have to calculate. For example, 8 windows. So it's hard to expect that the physician will sit down and perform the dosimetry as a routine. So good softwares are the softwares that are friendly and easy to use. Recently, we've seen several new software tools and better methodology to calculate the absorbed dose. Recently, the SNMMI Committee on Medical Internal Radiation Dose released the MIRD Calc and MIRD FI software. These are the products that are an initiative to provide useful dosimetry calculations to physicists. MIRCalc, MIRTFIT sheets are available free of charge at mirtsoft.org website. They are helpful to standardize methods to calculate the absorbed dose at the level of organ. All the calculations are made in Excel software, so such programs can be used to verify the dosimetry calculations provided by commercial software used in your nuclear medicine departments. We cannot compare the commercial software. Usually they are closed. Sometimes they are not described in detail, but we can compare the results of measures, that is the absorbed dose or the activity measured in the void, which is the view of interest. There's also an idea of developing a testing object, digital testing object. This will allow us to benchmark clinical dosimetry codes. It will be for the internal dosimetry. We are waiting for PET-CT. It is certain that standardization and harmonization of the tools for clinical internal dosimetry is a very important challenge to nuclear medicine. To end my lecture, another quotation from the publication of Manuel Bardies. There are no doubt many challenges ahead, but as dosimetry becomes accepted into routine clinical practice, the increased infrastructure required to implement it will only benefit the field of nuclear medicine. Molecular radiotherapy presents the unique capability to follow the fate of the drug administered to the patient. It allows the calculation of an objective index, and this index is the absorbed dose. To calculate this index provides us with a deeper understanding of the effects of the therapeutic procedure. This is too good an opportunity to miss. To sum up my lecture, I would like to say, firstly, personalized dosimetry is a must, but it is technically difficult and requires specialized software and well-trained professionals for the clinical implementation. Secondly, commercial software purchase sometimes can turn into a black box. Before clinical implementation, it requires proper calibration, proper understanding and adjustment to imaging scanners. Thirdly, phantom measurements are needed to verify the correctness of the obtained dosimetric reports. And finally, there is a need to standardize and harmonize the tools for clinical internal dosimetry. Here you can see the photo of the place where the source materials were found. I took part in a training in Houston. The training was held in a beautiful building with a great architecture. The interior design was also incredible. This building was almost empty because after COVID pandemic, most of the personnel are working from home. So thank you for your attention.